Hi, how are you doing today? This is Dr. Hamid, an ECPMG certified physician. We have a very high yield revision for the whole biochemistry chapter for the USMLE Step 1 exam. First is the DNA structure and organization. The DNA has double helix structure, so this means it has two complementary strands. One is running from 5' to 3', and the complementary strand is run at the opposite direction from 3' to 5'. Next is there is nucleotides that form the DNA. So the nucleotides are composed of deoxyribose sugar, phosphate group, and one of the nitrogenous bases, either A, T, G, and C. So adenine, cytosine, guanine, and thymine. So the adenine and the thymine are bonded together, while the cytosine and the guanine are bonded together. So A, T, and C and G. And the adenine and the thymine are bonded by two bonds versus the C and G, cytosine and the guanine, they are bonded by three bonds. So this translates to a different melting temperature. So the more there is an increase in the CG, which has three hydrogen bonds, the more the DNA melting temperature. So the CG content determines the DNA melting temperature, and the more we have CG bonds, the more we have a higher melting temperature for our DNA. Next is a chromatin structure. The DNA is very long and can't fit into the nucleus unless it has a special 3D structure. So the DNA is wrapped around histone octamers, and then we have like free DNA that is running, and then again DNA being wrapped around histone octamers, and so on. The free DNAs and DNA being wrapped around histone octamers. So this it's called the nucleosomes, and we have nucleosomes and the free DNA, and the whole structure is called the chromatin. So the chromatin is formed from DNA being wrapped around histone octamers, forming nucleosomes. So this is the chromatin. Next, the chromatin is further subdivided into euchromatin versus heterochromatin. So euchromatin is less condensed and it's transcriptionally active. So at the nucleus, we have like areas of heterochromatin and areas of euchromatin. The more we have euchromatin, the more the cell is means that the cell is transcriptionally active. By transcriptionally active, we mean it forms more RNA out of DNA. So the RNA being formed out of DNA is called the transcription versus the translation, which means the formation of proteins out of RNA. So euchromatin less condensed, more transcriptionally active, versus the heterochromatin highly condensed upon each other and transcriptionally inactive. And the next is the DNA replication basics. So we have some concept. The DNA replication is semi-conservative. What we mean by this, these are like the parent strands. This is a parent strand, and this other parent strand, and it is divided by an enzyme called helicase. And this is a complementary strand and also a new complementary strand. So each DNA daughter has one from the parent and one complementary strand. So it is semi-conservative by this way. It has each daughter DNA molecules contains one parental strand and one newly synthesized strand. Next is the direction. The DNA synthesis occurs from 5' to 3' direction. So we have two strands, one running from 5' to 3', the complementary strand running from 3' to 5'. So first, the strain of events starts with the enzyme topoisomerase, which breaks the outer bonds to make our smooth strands. For the helicase enzyme to cut it into two halves, two parents like strands, separate strands, and the DNA binding proteins, the single-stranded binding proteins. Its function is to prevent re-annealing. So after the helicase has done its job and like separated the parent strand into two strands, we have the single-strand binding proteins. These proteins prevent the DNA from re-annealing again. Next is that we have our main enzyme, which is the DNA polymerase. We have two enzymes. The first, DNA polymerase 3 and the DNA polymerase 1. The DNA polymerase 3 is the main replicative enzyme and elongates the leading strand and the lagging strand fragments. So this is the main enzyme. So this is the main enzyme. It has a special direction from 5' to 3'. So it acts continuously when it complements the 3' to 5' strand. So this strand is from 3' to 5', and the complementary strand is from 5' to 3'. This is why the DNA polymerase 3 here acts continuously versus the other side that is supposed to be formed from 3' to 5', and the DNA polymerase 3 can't do that. So it has to form it discontinuously. It forms something called Okazaki fragments, and the Okazaki fragments is bonded by an enzyme called the DNA ligase that is gonna bond Okazaki fragments together to so we have one smooth continuous complementary strand. Next You have to know that the DNA polymerase tree can't start from scratch. It has to build on something. So it has to start building on something called RNA primer. So there is an enzyme called primase. The enzyme primase is the one that's supposed to form RNA primer, and the DNA polymerase tree acts upon this RNA primer to form shorter Okazaki fragments at this strand and form one continuous DNA complementary strand on the other side. So here again, also it has to work on RNA primer. So after forming this RNA primer, we had to replace RNA primer by DNA. So this is the function of other DNA polymerase, which is DNA polymerase 1. So we have two DNA polymerases. The DNA polymerase 3 is the main replicative enzyme. That is the one that is doing the whole job. So, and the DNA polymerase 1 has a minor rule, which is the removal of RNA primers and replacing it with DNA. So this is the DNA polymerase 1 that has minor rule versus the DNA polymerase 3, which has the main rule here. So the DNA polymerase 3 forms the complementary strands, and then they are like ligated by DNA ligase. So this by this way, we're gonna form like two daughter strands. Next, the replication fork, which is a Y-shaped region, again, where DNA strands separate and the replication occurs. And this is the function of the enzyme helicase that is going to act as the replication fork to separate the two strands of our parent DNA. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously as Okazaki fragments. The origin of replication is a specific DNA sequence where replication begins, which is it is single in prokaryocytes and multiple in eukaryocytes. So we have to have like multiple origin of replication, and the DNA replication occurring at like multiple areas simultaneously. So we have like a very fast replication. This is true for eukaryocytes, so which have like which are more complex, and the origin of replication is single for prokaryocytes. There is something called AT-rich sequence. It is found in promoters and the origin of replication. So remember, the AT has two hydrogen bonds, so it is easy to be broken. So this way, this is why it is acts as a promoter regions and the origin of replication, such as Tata box regions. So this is due to like being easily broken compared to areas where the CG content with three hydrogen bonds versus AT areas with two hydrogen bonds more easily to be broken. So again, we're gonna go through the enzyme helicase. This unwinds DNA template at replication fork, and the primase creates RNA primers to initiate DNA synthesis. So this is a primase initiate. So for the Okazaki fragments, it acts on multiple sites. So form is the beginning of each Okazaki fragments. But here on the other side, we have one RNA primer. So this is the enzyme primase that is gonna create RNA primers. DNA polymerase 3 is the main replicative enzyme, elongates the leading strand and the lagging strand fragments from 5' to 3', while the DNA polymerase 1 removes RNA primers and replaces it with DNA. The enzyme ligase joins Okazaki fragments by creating phosphodiester bonds between different Okazaki fragments. Topoisomerase acts like on the outside of the whole structure to smooth and make the strand ready for the helicase to do its job. So the topoisomerase relieves the supercoiling by creating temporary breaks in the DNA strand, and the telomerase adds some DNA tags, TTA3Gs, to the 3' end of the chromosome. So with each replication, we may lose some segments. So the enzyme telomerase adds the tag sequence to the 3' end. So instead of losing our DNA, we're gonna lose some nucleotides from this tag sequence. By this way, we're gonna preserve the DNA. So the telomerase enzyme adds the DNA tag sequence to the 3' end in eukaryocytes. The DNA polymerase has 5' 3' synthesis and 3' 5' proofreading exonuclease activity. What does this mean? We have talked about how the DNA polymerase is the main enzyme is gonna form the complementary step from 5' to 3'. But what if we have some base pairing wrong? We it added a wrong base pair, so it is gonna act at a reverse direction to remove this base pair by something called the 3' 5' proofreading. So it's gonna proofread and do some exonuclease activity. It's gonna remove it. So it has 5-3- synthesis function, and 3-to 5- proofreading exonuclease activity. So the telomerase is upregulated in cancer cells and the progenitor cells. So this is the enzyme that is gonna keep our DNA from being lost. It is downregulated in aging and progeria. So we have like some drugs such as etoposide and etinoside. They are inhibit eukaryotic topoisomerase 2, irinotecan, topotecan, inhibit topoisomerase 1. Remember, topoisomerase is an important enzyme. Is gonna remove like supercoiling and break external bonds for the helicase to start. So this is an essential enzyme for the replication. So if we're gonna inhibit it, we're gonna halt the cell progression. So we what this equivalent to being a chemotherapeutic agent. So the hallmark of chemotherapeutic agent is halting cell progression and stopping the division. So if we're gonna inhibit topoisomerase 2 by etoposide, tinoside, or irinotecan, topotecan inhibiting topoisomerase 1, this is the way for chemotherapeutic agent to do its job. Next is the Bloom syndrome. The Bloom syndrome is caused by a deficiency in a helicase. So the helicase also has an important enzyme. So we've got deficiency in it. We have some condition called the Bloom syndrome, which has BLM gene mutation. The DNA repair mechanisms. So there are different mechanisms for DNA repair, either nucleotide excision repair or base excision, mismatch repair, non-homologous end joining, and homologous recombination. Nucleotide excision repairs means removing bulky helix distorting lesions, so such as pyrimidine dimers from UV lights. So we have some bulky helix distorting regions. So for example, here, these are like the UV light forming something called thymine dimers. So we have thymine dimers here, we're gonna remove it. So there is an enzyme called the nuclease that is gonna cut it here and cut here, and the helicase is gonna remove the damaged section, and the DNA polymerase 1 and the DNA ligase makes the repair. Remember, DNA polymerase 1 was the one that removed the primers and formed DNA instead of it. It has another rule here, which is the repair rule. So we have these pyrimidine dimers, and the nuclease is gonna cut far away, some base pairs far away, and the helicase is gonna remove the damaged section, and the DNA polymerase 1 and the DNA ligase is gonna make the complementary strand and ligate it. So this is the way we're gonna remove bulky helix distorting regions such as pyrimidine dimers from UV light. And this is defective in xeroderma pigmentosum. Remember, the people with xeroderma pigmentosum can't stand UV lights or the sunlight, because if you look at the starting trigger for this chain of event is the UV light. So the UV light is the one that formed thymine dimers, and that is like the one responsible for this error. So this is the nucleotide excision repair. Next is the base excision repair. Remove the single damaged bases. So instead of removing bulky helix distorting lesions such as nucleotide excision repair, so the base excision repair is different. It removes the single damaged bases. So it involves glycosylase that creates aburinic apyrimidinic sites. So aburinic apyrimidinic sites, we're gonna act at the level of the bases of cells. So we're gonna remove the purines and the pyrimidine bases. So this is another level of repair. And then the mismatch repair. So the mismatch repair is the way we're gonna correct mismatched nucleotides in the newly synthesized DNA. And this is defective in the condition such as Lynch syndrome, which has another name called hereditary non-polyposis colorectal cancer. So hereditary non-polyposis colorectal cancer, aka Lynch syndrome, has defect at the level of mismatch repair. So this is supposed to correct mismatched nucleotides in the newly synthesized DNA. So it detects and corrects slipped strand mispairing or the mismatched nucleotides. So this if affected we have Lynch syndrome. Non-homologous end joining versus homologous recombination. Non-homologous end joining reverse double-stranded breaks without requiring homology may result in loss of genetic information. This is defective in ataxia telangiectasia. So this is non-homologous end joining. We don't build upon another strand. It just repairs randomly, repair double-stranded breaks, doesn't require homology, may result in loss of genetic information, but it is gonna seal the break. So its main function is gonna seal the break. If we have defect in non-homologous end joining, we are proposed to ataxia telangiectasia. Versus homologous recombinations that uses a homologous DNA as a template for accurate repair. So non-homologous end joining is gonna seal the gap and may result in loss of genetic information, but homologous recombination uses homologous DNA template for acute repair. This is defective in a BRCA1, BRCA2 mutation, breast and ovarian cancer, and also in Fanconi anemia. So you have to remember with which goes with which. So the nucleotide excision repair removes the bulky helix distorting region and is defective in xeroderma pigmentosa, where the defect at the base excision level removes the single damaged bases. And the mismatch repair, you have to memorize it is like associated with Lynch syndrome, hereditary non-polyposis, colorectal cancer, mismatch repair defect. Non-homologous end joining goes with ataxia telangiectasia versus homologous recombination defect is with BRCA1, BRCA2, and Fanconi anemia. So BRCA1, BRCA2, Fanconi anemia, defective homologous recombination, non-homologous end joining ataxia telangiectasia, nucleotide excision repair, xeroderma pigmentosum, mismatch repair, Lynch syndrome. These are very high yield things to memorize. And the DNA mutation and the consequence, there are maybe silent mutations, missense, nonsense, frame shift, split size mutation, kind of DNA mutation and its consequence. So the mutation may be silent. So there are like 3 codons, first, second, and the third codon. So each combination of the 3 codons doesn't necessarily like code for a different amino acids. They are like amino acids can be produced by many different combination of codons. So if we got an error that is gonna give like same amino acid, so this error is gonna pass silently. So this what we mean by silent mutation. So this codes for the same amino acids. This often occurs in the third codon position. So if we got defect in the third codon position, we may end with like the code for the same amino acid, and there is no change in the protein, and everything is very good here. So this is the silent mutation. Next is the missense mutation. So the missense mutation means we've got a different amino acid. So the famous example for the missense mutation is glutamic acid being transformed into valine. So in the beta globulin of the hemoglobin, we got glutamic acid normally. If it is converted into valine by having a missense mutation and the difference in the codon resulted in a different amino acids, we will have sickle cell disease. This is an example of missense mutations. Next is the nonsense mutation. So the nonsense mutation is the creation of premature stop codon. So why this is like nonsense? The different codon, the error that resulted in the different codon didn't produce another amino acid such as in the missense mutation, but instead it produced the premature stop codon. So this is why it is nonsense. It doesn't produce another amino acid. It produced a codon that is gonna stop protein synthesis. So it creates, so instead of having nonsense mutation, instead of creating a different amino acid, it creates another thing, which is a premature stop codon that is gonna stop translation. So UGA, UAG, and UAA. So UGA and the opposite of it, which is UAG and UAA. So remember that. Results in truncated over non-functioning proteins such as Duchenne muscle dystrophy, some forms of Duchenne muscle dystrophy. So the silent mutation, no example because it doesn't do anything. Missense mutation, the famous example of sickle cell disease. Nonsense mutation, some forms of Duchenne muscle dystrophy, and our truncated non-functioning protein due to UGA, UAG, UAA. Next, frame shift mutation. So insertion or deletion of nucleotides not divisible by 3 is gonna be a very, very bad problem because each remember there are like 3 codons for each amino acid. So if we like, for example, add 3 codons, it's gonna shift the process and we will have the same sequence of the consequent amino acid. But let's suppose we have insertion or deletion of nucleotides not divisible by 3. So this is gonna like affect the whole consequent chain of amino acids. So this is gonna alter reading frame of all downstream codons. Example: Duchenne muscular dystrophy, Tay-Sachs disease, cystic fibrosis. So remember there are some forms of Duchenne muscular atrophy resulted from nonsense mutation, others resulting from frame shift mutation, and Tay-Sachs disease and cystic fibrosis. Another example so that involves frame shift mutation. Split mutation affects intersplicing, often retaining introns in mRNA. Remember mRNA like has introns and exons. So if it retains introns, so this is an example of a splice site mutation such as beta thalassemia. The beta thalassemia sometimes called by splice site mutations. So the degree of functional mutation, so the silent is the least affected. Next is the missense mutation. So it's gonna produce another amino acid such as sickle cell disease. Next, nonsense mutation is gonna cause truncated like form of protein such as Duchenne muscle atrophy. And the next is a frame shift mutation, the most dangerous because it affects all consequently synthesized amino acids. So examples, Duchenne muscular dystrophy, Tay-Sachs disease, and cystic fibrosis. So the Duchenne muscular dystrophy, again, can be caused by nonsense or frame shifting mutation, while Becker muscular dystrophy caused by some other thing called in-frame deletions. Next is different kinds of RNA polymerases. So we have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So we have three different RNAs, rRNA, mRNA, tRNA, and the 5S rRNA. So the RNA polymerase 1 is gonna synthesize ribosomal RNA, rRNA. 2 is gonna synthesize the messenger RNA, mRNA. 3 is gonna synthesize the tRNA or transfer RNA, and the 5S rRNA. So the location, the RNA polymerase 1 is located in the nucleoli. So the ribosomal RNA synthesized in the nucleoli. So this is the most central. So RNA polymerase 1, ribosomal RNA, most central in the nucleoli versus the RNA polymerase 2, messenger RNA, and the transfer RNA way out in the nucleoplasm. And the rRNA is the primary component of ribosome. So this is like the protein factory. The ribosome rRNA is the primary component. And the RNA polymerase 2 synthesizing rmRNA. So it binds to TATA box, TATA box with transcription factors. So it is inhibited by alpha amantine in this cab mushrooms. So it's like you have to know this example that alpha amantine in this cab mushroom is the one that's gonna inhibit RNA polymerase 2. And the RNA polymerase 3 is like the one that's gonna synthesize the tRNA and the 5S RNA located in the nucleoplasm. tRNA delivers amino acids during the formation of the protein is our like transporting vehicle is the tRNA. So the different types of RNA polymerases, RNA polymerase 1, RNA2, polymerase 2, and 3. So the RNA polymerase 1 is the one that is going to synthesize ribosomal RNA. So we have ribosomal RNA, messenger RNA, tRNA, and the 5S rRNA. The RNA polymerase 1 is the one responsible for ribosomal RNA synthesis. 2, messenger RNA. 3, transfer RNA, and the 5S rRNA. So the location, RNA polymerase 1 located in the nuclei. This is the most central in the nucleoli, while 2 and 3 located in the nucleoplasm. And the RNA polymerase 1 synthesizing RNA, so the ribosomal RNA is like the factory for like proteins, so it's the primary component of ribosomes, versus RNA polymerase 2 forming mRNA, so the mRNA. There is RNA polymerase 2 plus transcription factors binding to data box within the promoter sequence. Now, if the data box is mutated, so this is gonna mean the decreased binding of RNA polymerase. So this is the function of RNA polymerase 2, and it's like rule of synthesized messenger RNA. There is it is inhibited by alpha amantine in this cab mushroom. So the alpha amantine is gonna like inhibit RNA polymerase 2 versus RNA polymerase C, which is responsible for the synthesis of the transfer RNA. This is like the vehicle that delivers amino acids during the translation. So the ribosome is the main factory, and the tRNA or transporter RNA is like the vehicle that is gonna deliver amino acids for translation, aka protein synthesis. So the mRNA processing in eukaryocytes, so the initial transcript of mRNA called heterogeneous nuclear RNA. So this is like the initial transcript, and it has to be acted upon. So the first there's addition of 5-methyl guanosine cap. So at the 5- end, we will add 5-methyl guanosine cap. On the 3- end, we're gonna add poly A tail. So this, of course, it was a heterogeneous nuclear RNA, which is like the initial transcript. Then we're gonna splice introns. So it's gonna splice and remove introns, and it's gonna export the mature mRNA to the cytosol for translation. So all of this mRNA processing occurs like in the nucleus before being transported into the cytosol. First, addition of 5-methyl guanosine cap and 3- poly A tail and splicing of introns, then extrusion into the cytosol for translation. So this is the RNA polymerase. So binding at a tata box. So this refers to RNA polymerase. So RNA polymerase that is gonna form is RNA from DNA. And these are the deoxynucleotides. Then this is the enzyme RNA polymerase, and this is a DNA RNA hybrid. This is RNA being formed out of the DNA. Here, this is the direction of polymerization, and this is the coding strand. And of course, there is unwinding of DNA. So the regulation of gene expressions. So we have some regulatory elements as the promoters, the enhancer, silencer, DNA methylation, and the histone modification. The promoters, they are like the sites where RNA polymerase 2 and the transcription factors behind it. Remember, RNA polymerase 2 is the one that is responsible for forming messenger RNA. Remember, it binds at some like sequence called Tata box or cat box, aka the segments containing AT rich sequence. So the promoters are located upstream from gene locus. So it doesn't mean it like at the start of gene locus, it occurs upstream of it. So this is the promoters, the site with AT rich sequence, the sequence with 2 hydrogen bonds that is easy to break and to start replication from this sequence. So this is are the promoter regions upstream from the gene locus, the site for RNA polymerase 2 and the transcription factors to act. Next is the enhancers. These are the DNA regions that upregulate gene transcription. So can be located far from the genes they regulate and bound by activator proteins. So these are like enhancers. So they are maybe very distal to the regions that is gonna enhance, not need to be near. So they upregulate gene transcription. They bound by activator proteins. So these are like the activator proteins. So these are the activator proteins that is gonna bind to enhancer region. So first you have to know they may be like far away. Next is they are bound by activator proteins that's gonna affect like maybe distant gene regions and enhance their like transcription. And the silencers, they are like DNA segments that down regulate on the other hand gene transcription. They are bound by repressor proteins. So the enhancer bound by activator proteins versus the silencers that are bound by repressor proteins. The DNA methylation means the addition of methyl groups to DNA, typically as a CBG island. They generally replace gene transcription. So this refers to like epigenetics. This is a field where there are like control of the genes without changing its sequence through methylation or acetylation. So the DNA methylation generally represses gene transcription. We don't like change the sequence, but we had methyl groups typically as a CBG islands. So this involves genomic imprinting and X-chrome inactivation. So this switches on and off without changing the gene sequence, and histone modification. So the histone, remember, the chromatin had like stones and the DNA wrapped around it. So by this way, by manipulating the histone, we're gonna like control the proliferation of the cells and the replication. So the acetylation relaxes the DNA coiling and increases transcription, which is the formation of like RNA. And the deacetylation ties the DNA coiling. So the deacetylation is activatory, so relaxing and making the transcription increase, while deacetylation tightens coiling and it's inhibitory. It's gonna decrease transcription. Methylation, of course, is gonna suppress transcription. Remember DNA methylation or methylation of histone. Any methylation here is gonna suppress transcription versus acetylation, which is activatory. So it's gonna activate it by relaxing the DNA coiling. So they remember euchromatin transcriptionally active, less condensed. So we have increased the histone acetylation. Remember, it's activatory versus the heterochromatin highly condensed, transcriptionally inactive, has increased the methylation with decreased acetylation. Enhancers can be located like hundreds or thousands of bases away from the genes they regulate, and like the dysregulated DNA methylation is implicated in fragile X chromosome syndrome. So this related to like switching on and off of expression. So this is related to fragile X syndrome, and altered histone acetylation may contribute to gene expression changes in Huntington disease. So you have to get a paper and like write every like syndrome and the change that is like related to it. So Huntington disease is related to the acetylation of histone versus like DNA methylation dysregulation is related to fragile X chromosome. So remember, here's this enhancers and like these like the activators that is gonna bind to enhancer and affect like areas that is gonna genes that may be 1,000 bases away from it. And this is a promoter, which is the area rich in T80 sequence. So the DNA polymerase and all the replication is gonna start from the promoter regions. The clinical application, xeroderma pigmentosum, remember, defect in the nucleotide excision repair related to UV. So the UV lights is the thing that is gonna like started the cascade of events. So the UV causes induced by pyrimidine dimers, and if we have got a defect in the nucleotide excision repair, we will have xeroderma pigmentosum. So the clinically will have extreme photosensitivity and early skin cancer, dry skin. Next, Lynch syndrome, defect in the mismatch repair genes MLH1, MSH2, MSH6. So this is related to microsatellite instability. So there is an increase in the risk of colorectal, endometrial, and other cancer. You have to memorize colorectal cancer and endometrial cancer and Lynch syndrome. And also the defect in the microsatellite instability. So the sickle cell disease, missense mutation, glutamic acid is transformed into valine in the beta globulin. So this results in abnormal hemoglobin S. So these patients will have vaso-occlusive crises, hemolytic anemia, and susceptibility to different kind of infection with special immunization schedules. So fluoroquinine toxicity. So the fluoroquinine mechanism of action is the inhibition of prokaryocyte topoisomerase 2. So remember, it's gonna inhibit like replication for these organisms. So this is the way it's gonna fight bacteria, but it may have like side effects by inhibiting eukaryocyte cells. By this way, it's gonna cause tendonitis or tendon rupture by like killing our cells. So Achilles tendon rupture, very famous, famously associated with fluoroquinolone toxicity. So it cannot be taken with the divalent cations, calcium, and the iron. You have to watch for this if you have if you are prescribing one of them. Comment with if you are prescribing one of them with fluoroquinolone. So DNA replication semi-conservative occurs from 5' to 3', Okazaki fragment joined by DNA ligase. There are stop codon UGA, UAG, and UAA. So these are the stop codon. And if we have like nonsense mutation, we're gonna get one of these, and we will have truncated protein. The alpha amantine like inhibits RNA polymerase 2. That this is the one that is responsible for our messenger RNA. So I hope you guys enjoyed this episode, and see you in next episode to continue our high yield biochemistry revision. Thank you.